So, what is biofertilizer? Now, as the word suggests, bio, also known as natural fertilizers. They are microbial inoculants. I suppose you know the meaning of inoculant. Inoculant is the total number of microbial cells, whether bacteria or fungi or algae, whatever you are taking for some useful purpose in the microbiological operations. So, biofertilizer is a natural fertilizer that are microbial inoculants of bacteria, algae, and fungi, prepared separately or in combination. So, these biofertilizer helps in biological nitrogen fixation, or it helps in the non-soluble form of phosphorus to be making it available for the solubilized phosphorus to be formed, which effectively is being utilized by the benefits of plants. So they build up the soil microflora, thereby increasing the soil health. So they are also act as a conditioners. Biofertilizer also includes organic fertilizers like manures, etc. So use of biofertilizer is always recommended by agricultural microbiologists for improving the soil fertility, or for improving what's also known as organic farming nowadays. So benefits of biofertilizers that they are eco-friendly, that is, they are not synthetic chemicals, so they are friendly to the ecosystem. They increases the soil fertility. It has also been observed that it increases the crop yield. So yield, net yield of the economically important part of the plant is increased. They are low cost and easy to grow, so you don't require an input of very high end equipments and all those things for its growth and production. Even farmers can also grow blue green algae and azula in their cropping areas. And of course, biofertilizer makes the resistant to plant pathogens. So many of the plant biocontrol agents nowadays has been developed, which makes the plant pathogen resistant. Now, in biofertilizer, they are divided into nitrogenous fertilizers, phosphate solubilizers, mycorrhizas, and green manuring. In the case of nitrogenous fertilizers, symbiotic nitrogen fertilizer like rhizobium, and non-symbiotic like Azotobacter, Azospirillum, Cyanobacteria, and Azula. In the case of phosphate solubilizers, many of the bacteria and fungi like Pseudomonas and Micrococcus and Aspergillus species are known to degrade the insoluble form of phosphorus into soluble form. And then you have green manuring, in which you add some proteinaceous plant like Sesbania aegyptica and Lethira sativus and Tephrosia purpurea into some uh, paddy manure or some farmyard manure, which increases the nutrient availability of the protein content in the soil, which hereby affects the plant growth. In the case of symbiotic nitrogen fixers, many of the symbiotic nitrogen fixation occurs between leguminous plants. So leguminous is a family of family Fabaceae, has many of the plants like Medicago sativa and pea plants and groundnut. They have in their root nodules some rhizobium being growing. Now this rhizobium bacteria in laboratory can be cultured on the Yema medium, which has dipotassium hydrogen phosphate and magnesium sulfate and sodium chloride, mannitol, and yeast extract in their medium, and you can isolate the rhizobium bacteria from the root nodules of these leguminous plants. Asymbiotic nitrogen fixers are nitrogen, it converts nitrogen into ammonia in the soil itself. So these bacteria like Pseudomonas and Cyanobacteria, Azotobacter, Azospirillum, they actually does not fix it in the root nodules, but they directly convert nitrogen into ammonia. Some phosphate solubilizers like Pseudomonas, Bacillus strains, and Micrococcus, and some fungi like Aspergillus. They are grown onto Picosla slovakia's medium. So Picosla medium is in which you can grow these bacterias, and they can convert the insoluble form of rock phosphates into soluble forms of rock phosphates. These microbes are, as the name suggests, that these bacteria can convert into insoluble to soluble form. The role of biofertilizer in agriculture is very multifaceted kind of role. If you see, it has broad areas to explore in terms of nutrient signaling, in bioremediation, in biocontrol, in biofertilization, and in phytostimulation. In the case of bioremediation, it cleans up the contaminated soil, and in the biocontrol, these fungus like trichoderma and all of the actually reduces the pathogens, and it reduces the disease management of the particular pathogenic fungus, and it gets a biofertilization. It results into nitrogen fixation and phosphate solubilization. In phytostimulation, it goes for production of IAA and several ACC deaminase enzymatic activity. So this is a kind of an eco-friendly approach in the field of agriculture, which increases the fertility of soil in short way and as well as in the longer way. Now to the microbiological part of microbiological instrumentation part of the biofertilizer technology, how you can isolate and how you can cultivate for the large scale production. We all know that the capacity of these microorganisms to have their beneficial effect, but how we can isolate them and how we can cultivate them in the instruments, in the microbiological instruments. So for the isolation of bacterial biofertilizer from nodules, you should have the root nodules of a family of Leguminosauridae, and then you should wash it for 3 minutes, and then you can sterilize it with 0.5% of sodium hypochlorite for 30 seconds, and then you can dip it in 70% ethanol for 1 minute. So this dipping in sodium hypochlorite and ethanol will kill all the pathogenic bacteria into it, and it will make the surface of the root nodule completely sterilized. And then you can dry it with sterilized filter paper, and then nodules are crushed in sterile buffer, and then you can inoculate in the medium, and within overnight you will see, next day you will see the growth of rhizobium in the culture of the petri plate. Now when it comes to mass scale cultivation of the bacterial fertilizers, now from the mother culture which we have isolated in the last slide, we have seen how to isolate. Once you maintain the mother culture means original culture of the bacteria. Then you may prepare a slant out of it, and then you can make a broth. Broth is in which you don't have agar-agar medium. Now this broth is cultured in a small flask, and then this broth is transferred in the big flasks or in bottles, and these shakers are limited requirement are being stirred, and then this broth is blended with sterilized carrier peat or lignite. So we'll be seeing in forthcoming slides that what is the carrier in the case of large scale production of biofertilizer. So in this carrier molecule, which is known as peat or lignite, these are packaged in the polythene bags, and then they are stored at 25 degrees centigrade. And this peat culture quality checking is done that it should have only the beneficial microorganism. It should not have the pathogenic organisms in this peat culture, because this peat culture is the culture which the farmers would be using, and it would be introducing in the soil for enhanced fertility. Okay. So they are stored for 4 degrees centigrade in lower temperature, and then they are dispatched to or marketed to the farmers.